Good morning. My name is Simone Panigada. I am the president of the Tethys Research Institute, an NGO based in Italy for the conservation of cetaceans and marine mammals in the Mediterranean. And I chair the scientific committee of ACOBAMS, an international agreement for the conservation of cetacean in the Black Sea, the Mediterranean, and adjacent Atlantic waters. Today I will present you some inputs on conservation policy, how to use scientific knowledge to support the decision-making process, sort of a path from science to policy. I will talk about the Convention on Biological Diversity and on the EPSAS in particular. Then I will be talking about the IUCN, important marine mammal areas and key biodiversity areas. And then I will briefly touch upon BirdLife International and the international important bird areas. In 2006, in response to the Johannesburg Plan of Implementation call to establish protected areas globally, the Convention on Biological Diversity started to develop and apply criteria to describe and designate EPSAs, which are ecologically of biologically significant marine areas in the world's ocean, which will, inter alia, support the nation's 2010 commitment to achieve the Aichi biodiversity targets by 2020. And I will briefly talk about the Convention on Biological Diversity in a different presentation. The EPSAs are special marine areas that serve important purposes to support the healthy functioning of oceans and the many services that they provide. In 2008, the CBD adopted 7 scientific criteria for identifying EPSAs in need of protection in open waters and deep sea habitats. These criteria are uniqueness or rarity, special importance for life history of species, importance for threatened, endangered, or declining species and or habitat, vulnerability, fragility, sensitivity, slow recovery, biological productivity, biological diversity, and naturalness. The process has been applied worldwide and in the Mediterranean Sea as well, and this was done through a series of workshops, and the final one was in Malaga from 7th to 11th of March 2014. Here you see the whole delegates and experts invited, and the discussion that followed was very important because it allowed to create quite a few EPSAs in the Mediterranean Sea. This is a map with an area that meets the EPSA criteria in the Mediterranean Sea. As you can see, some of them are very wide, for example, in the northern Mediterranean Sea, in the Strait of Sicily, in the Gulf of Sirte, and Libyan waters, Egypt. So they can cover a wide surface of ocean. Similarly to the EPSA process, there is another ongoing conservation effort, which is led by the Marine Mammal Protected Areas Task Force, which is established under the IUCN, International Union for Conservation of Nature, with the Species Survivor Committee and the World Commission on Protected Areas. And this is called the Marine Mammal Task Force. And the goal of this is to contribute to place-based marine mammal conservation through the establishment of important marine mammal areas, or IMAs as they are called. The task force has a dedicated website, which is marinemamalhabitat.org, where all the information that I will be presenting are available, and all the workshop reports, as well as an e-atlas with all the information related to each important marine mammal areas are available. What are IMAs? IMAs are place-based conservation tool identifying discrete portions of habitat important for one or more marine mammal species that have the potential to be delineated and managed for conservation. IMAs are not marine protected areas, and this is a very important concept, and are not identified on the basis of management considerations and criteria. The identification of IMAs is an evidence-driven, purely biocentric process based on the application of scientific criteria and on the best available science. And we will go through this in more detail throughout my presentation. Marine conservation and management initiatives which can utilize a product of the IMA processes are, for example, the CBD through the EPSA process, which we just presented, marine spatial planning and management of any human activity at sea, for example, shipping, fishing, industrial and scientific exploration, the establishment of marine protected areas, MPAs or MPA networks, the International Maritime Organization, IMO, through PSSA, for example, particularly sensitive sea areas, or the IUCN standard key biodiversity areas. A very important tool that is facilitating the IMA process is the Resolution 1213 from 2017, which acknowledges the IMA criteria and process and requests CMS parties and invites range states to identify specific areas where the identification of IMAs could be beneficial. This was adopted in Manila a couple of years ago. The IMA identification process follows 3 stages. Stage 1, called the areas of interest or AOI, where collected information indicating areas of interest, areas of suitable evidence are collected. Stage 2, candidate IMAs, regionally proposed and accepted by expert workshops. And then stage 3, official IMAs, globally important areas peer-reviewed and accepted by an external panel. So the stage 1 is led by data collection. Stage 2, we may need some new analysis, for example. And then stage 3 is advocacy. We have an IMA and we need to make sure that this is properly managed. The IMA work program between 2016 and 2021, we started with the Mediterranean Sea, which has now been done. And then we have done a series of workshops covering the southern hemisphere, and we still have 2 more to go. One, Australia and New Zealand in blue lines, and one in the Pacific Ocean west of South America and Mexico. Each workshop followed a predefined process, which has been developed in consultation with the regional marine mammal science and conservation community to identify candidate IMAs or CIMAs on the basis of received proposal for areas of interest. After the workshop, candidate IMAs are submitted to an independent review panel of experts to verify that the criteria were applied correctly and that candidate IMAs were identified on the basis of robust scientific information. This is very important. We need to base our selection on robust scientific data. Candidate IMAs are then converted into IMAs and are made available through the website and the dedicated IMA e-Atlas, which you can find on this link. This is how the IMA e-Atlas look like. We have IMA in orange, in a darker color we have the candidate IMAs, and in blue we have areas of interest. By clicking on any area, we access a summary, for example, in this case in the northwestern Mediterranean Sea, where a few information are provided. Clicking for more information, we access a PDF file with more detailed information such as area size, qualifying species and criteria, marine mammal diversity, and all the details that justified this IMA. And there are figures, there are references, bibliographic documentation, and the justification of each criteria. And then there is also an annex with supplementary maps if this can be helpful in providing more information on a specific area. Going to the different criteria in a little bit more details, we have criterion A, species or population vulnerability. Criterion B, distribution and abundance. This is subdivided in different sub-criteria: small and resident populations, aggregations. Criterion C, key life cycle activities, reproductive areas, feeding areas, migration areas. And criterion D, special attributes, distinctiveness and diversity. A little bit more details on each criteria. You can find these on the documentation which has been provided. There is a criteria document which is fairly complete. So criterion A, areas containing habitat important for the survival and recovery of threatened and declining species. And there is a advised numerical threshold, which is not applied to this specific situation, but it provides similar information related to the IUCN Red List. For example, whether they are critically endangered, or 0.5% of the global population, just to give some quantitative information on each criteria. Criterion B, distribution and abundance. B1, areas supporting at least one resident population containing an important portion or proportion of that species or population that are occupied consistently. And again, the numerical threshold advised. B2, aggregations, areas with underlying qualities that support important concentration of a species or population, and the relative advised numerical threshold is provided in red. C, key life cycle activities. We have C1, reproductive areas, areas that are important for a species or population to mate, give birth, and or care for young until weaning. And again, the threshold. C2, feeding areas, areas and conditions that provide an important nutritional phase on which a species or population depends. These are pretty much areas where the species we are considering is actually feeding and foraging. And the threshold. C3, migration areas, areas used for important migrations or other movements, often connecting distinct life cycle areas or the different parts of the year-round range of a non-migratory population. This is a little bit more complicated. For example, we have a population that meet in a place for feeding and then mate in a different place. They go through an area which is interested by the migration, and this is where we should establish an IMA. And again, the threshold. The last criterion is D, D1, distinctiveness, areas which sustain populations with important genetic, behavioral, or ecologically distinctive characteristics, with a threshold. And diversity, areas containing habitat that supports an important diversity of marine mammal species. And this can, of course, change and range between different areas. In the Mediterranean Sea, for example, the diversity is fairly low. We have about 10 or 11 species of cetaceans. In other areas, we can have up to 30 or 40 species, so the diversity index can vary pretty much depending on the area. And these are the numerical threshold related to these specific criteria. The process: before the workshop, we collect areas of interest, inventory of knowledge, data appraisal form to facilitate our knowledge assessments. During the workshop, we review the areas of interest submissions. We assign candidate IMA's writing group. The groups then meet to draft the submissions template, and then we agree on a final list of candidate IMAs. And this is what happens during a workshop. This is the template for drafting. And after the expert workshop, we send all the candidate IMAs to an independent review panel. We can decide to request for necessary correction, request for additional research, and an IMA is either established or not. The IMA delineation, we have a guidance book which is available online and as an additional documentation for you, where you can really read this in much detail. We need to provide evidence of a specific area, then we create an envelope around the area. Maybe there are more than one species, or maybe there is more than one criteria, and then we create a buffer to make sure that we consider overlapping habitats as well. For example, in this case, we have species A in blue, then you have species B in green, and species C in yellow. We can merge all of them and have a multi-species boundary which includes all the 3 species in a larger IMA, which of course will act as a protection area for more than 1 species. And we can have a core for 1 species, for example, and a little buffer inside that. Similarly to IMAs, we have important bird areas, which is what we used as a template to establish our area. An important bird and biodiversity area, IBA, is an area identified using an internationally agreed set of criteria as being globally important for the conservation of bird populations. In the IMAS concept, we are focusing on marine mammals. In this case, it is clear that we are focusing on bird only. IBA was developed and sites are identified by a company, by an organization, which is BirdLife International. Currently, there are over 12,000 international important bird areas worldwide. These sites are small enough to be entirely conserved and differ in their character, habitat, or ornithological importance from the surrounding habitats. In the US, for example, the program is administered by the National Audubon Society, which has been studying birds since many years, and they have developed the necessary and needed expertise to facilitate this process. Often, IBAs form part of a country's existing protected area network, and so are protected under national legislation. This is very important, otherwise, like IMAs, they are not MPAs, they need to be transformed into something which is legally binding and legally protected. Legal recognition and protection of IBAs that are not within existing protected areas varies within different countries. Of course, different countries have different legislation process. Some countries, for example, have adopted a national IBA conservation strategy, whereas in other, protection is completely lacking, and this is where advocacy, which we mentioned earlier on, is very important and can facilitate strengthening this process. The criteria for IBAs are determined by an internationally agreed set of criteria, and to be listed as an IBA, a site must satisfy at least one of the following rating criteria. A1, globally threatened species. The site qualifies, if it is known, estimated, or throughout, to hold a population of a species categorized by the IUCN Red List as critically endangered, endangered, or vulnerable. This is very similar to our IMAS concept. A2, restricted range species. The site forms one of a set of selected areas to ensure that all restricted range species are an endemic bird area or a secondary area and are present in significant numbers in at least one site and preferably more than one site. Biome restricted species. The site forms one of a set selected to ensure adequate representation of all species restricted to a given biome, both across the biome as a whole and for all its aspects in its range state. And the A4 is congregations, which is where several species of birds congregate. To finish, I am presenting a list of selected documentation and references, which can be accessed online, and they can provide some guiding tools and guiding material to better understand and better study this process. The criteria are well defined, and a link to the eAtlas where all the PDFs can be found is also presented.